Hi, everybody. So today we're going to talk about the scientific method. Now, science is about observing the world and making sense of it, and then often making predictions about the world and what you can expect based on your past observations. One of the tools that is used is a very formulaic process, controlled experiment, that is part of the scientific method that scientists can use. And so we're going to talk about that today. And we're going to start with the first thing the scientist does is goes out in the world and is curious about something. There's a question, something they're curious about. We call this the problem. The problem is often just something you're wondering about, a question. And it's not necessarily something you can test. You know, is something good or bad? You can't test goodness, right? You can test if something is good for growing tall, right? But you can't just test goodness, right? That's not testable. So a problem does not have to be something specifically you have to test. It's really the thing, the essential thing you want to know. The example that we're going to use today in the video is the question, do plants like music? Do they like music? Now, that's not testable. We don't have a way to measure how much plants like something. Right? That's absurd. So to be very careful, this is a problem question, not a hypothesis. And we'll talk about what that is a little bit in a few moments. Okay? So the first thing you're going to do is you're going to define the problem. Now, do plants like music? How do we figure that out? How do we figure out if plants like music? Well, put plants in a room and play music for them. Will that do it? No. We don't know what we're measuring because we don't know how to measure liking. When plants like things, we don't know how to measure that. And if we play music for plants, and let's say we have a specific thing we're going to see if they respond, like growing tall. Let's say we want to know if plants grow tall when they're given music. If we put a bunch of plants in a room and we play music for them and they grow, will we know it was the music? No, we need a basis for comparison. So in controlled experiments, we will have at least two groups of subjects in the experiment. And here I've shown two groups of plants, or two plants in this case, and we're going to treat them differently. We will take a group of them and play music, right? And we'll see what happens to them. But we're going to have a baseline, something to compare them to, and that's the other group. That group will not get music. And so we'll be able to say, you know, look, if we didn't play music for this group and we did play music for that group, this group grew more, it must have been the music, right? So that's how we do a controlled experiment. We have two groups, one gets something, one doesn't. That thing that we make different between the two groups is the thing we're testing. And then, you know, we'll look to see if they grow taller. If they grow the same amount in both groups, then we can say the music had no effect. If the group with music grows taller, then we could say, wow, it looks like music makes plants grow taller. And then that problem we were trying to get at, do plants like music? Well, you know, they grow taller. So that's kind of getting at what we really wanted to know. Okay, so that's how we would do the controlled experiment. Now, this will only work if we meet certain conditions. Let's say the group that's in the room with the music is also getting more sunlight. And the plants without the music are in a slightly darker room. And the plants with the music grow taller. Can we conclude that it was the music? No, it's probably that they're getting more light, right? So we have some other issues here that we have to deal with. We have to make sure that everything is absolutely the same between the two groups. They have to be the same species. We can't have one type of plant here and another type of plant here. One might be slower growing as a species, so we can't use that. We have the same species. They have to be given the same amount of water. We can't have one group have no water and one group have water, because then it will be the water that makes the difference in the outcome and their heights, right? We have to have the same soil, the same light, and so all those things have to stay the same. Now, then over time, we'll measure their height and we'll see if the ones with music grow taller. And if they do, we can conclude that music will make them grow taller. So this is a controlled experiment, and there's certain pieces to it that we are going to assign their role. So we have two groups of plants. We have the group without the thing we're testing, in this case, the music. It could have been water. It could have been a number of things that we were testing on plants. But the music here, we have a group of plants without music. They're the control group. We have a group of plants with music. That's the experimental group. Okay, so the control group, the experimental group. The thing that's different between them, you may have learned in the past, is called a manipulated variable, or simply the variable. It's the one thing that's varied or differs between the two groups, right? No music, music. Everything else stays the same. It's the variable, the independent variable. We won't call it the manipulated variable in biology. We're going to call it independent, because there actually turns out to be another variable. Now, this is confusing because I just said there's only one thing that's different between the two groups, and that's true. Only one thing different between the two groups, and that's the music. That's the independent variable. But we are predicting that if the music is different, something else will be different as a result. And what do we think will be different depending on whether they got music or not? Their height. So their height, we're not making different between the two groups. They'll start the same height, but whether they grow taller or not is going to depend on the music. So the music is the independent variable, and their height will depend upon that. At least that's what we're predicting. Okay, so we have the control group without the independent variable, the experimental group with the independent variable, and during the experiment, we are going to measure the dependent variable. And in the process of all this, we have to make sure that all those things in the middle are the same species, the same water, the same light. Those are called constants. Caution, lots of students call constants controls. They aren't. They are constants. The control group is the actual specimens we're testing that lack the thing, the independent variable thing we're testing. Okay? So the control group is the group of plants without music. The constants are all the factors that are the same between the two groups. Now, some states, actually, the curriculum calls these controls. So in New York State, under the New York State regions, we are going to call these constants, and this is the control group. But if you're watching this from another state, you may have different vocabulary for that. All right, so we're going to predict what happens. Now, what's your prediction? Well, you may be predicting that it's ludicrous to think that music would affect plants. They don't have ears. What are we talking about here? And you may say, wow, it doesn't matter. If plants get music, they're not going to grow any taller. So you may say, if plants are given music, they're not going to grow taller. You may say, wow, I actually read something about this. I heard something about this. It must be something about the sound waves hitting on their bodies, something. I think they're going to grow taller. So if plants are given music, then they'll grow taller. Both of those things are called a hypothesis. They are both correct hypotheses. We'll find out when we run the experiment if, in fact, the hypothesis is correct. But in terms of the way that the hypothesis is written, you can make a bad guess or a good guess, and it's still a good hypothesis. I want you to look really carefully. Now, in earlier grades, they always told you that the hypothesis should have an if and a then, and that does make it easier. It actually can be technically correct without that. As long as you're predicting an outcome that is measurable, it's a hypothesis. So you could just say plants that hear music grow taller. That would be a perfectly fine hypothesis, but please try to put it in an if-then format, and here's why. It's helpful for this other thing. Students frequently confuse the independent and dependent variables. If you can get your hypothesis written correctly, it will help you figure out what the independent and dependent variables are. This is why. If plants are given music, music here, was that the independent variable or the dependent variable? It was the independent variable. Turns out the if part of the statement always has the independent variable in it. The then part has the dependent variable. Then they will grow taller. The height is in the dependent, the then part of the statement. Okay, so if plants are given music, independent variable, then they will grow taller, dependent variable. So if you write the hypothesis in the if-then statement, you can then go back and check to make sure that you have, in fact, identified the independent and dependent variables correctly. So in our notes, we're going to very quickly get this all into the notes. And so let's look at this. So formulate a hypothesis. It cannot be a question. It's the problem that's written as a question. It cannot be a question. And in fact, on the regions, they will mark it wrong if you put a question mark, even if it's written like a statement. Or if you don't have a question mark, but it sounds like a question, it will be wrong. Try to put it in the if-then statement and you will never get it wrong. It has to be predicting an outcome. You're not asking a question about the world. You are predicting what is going to happen in your experiment, and it must be measurable. Don't say plants will do better. Do better? What the heck is do better? They will grow taller. Then you know exactly what you're doing. You're going to be measuring their height. Okay. So it has to be measurable. And in this case, if plants are exposed to music, then they will grow taller, was our example. All right, so we're going to design the experiment. Once we've predicted the outcome, we are going to have the basis for comparison. What did we call that? That was the control group. And that was the plants without music in this case. And then we're going to have a group that is given music called the experimental group. The thing that's different between the two groups, what did we make different? The music. That's called the independent variable. That's the thing we originally make different. Here, music. So one group without music, one group with. Independent variable. Then during the experiment, we're going to measure something that we think may be different between the two groups, depending on whether they got the music. That's the dependent variable. And here, that was height. It didn't have to be. We could have measured other things about the plants. We could have measured how many flowers they produced, or we could have measured their mass, or how green their color was. There's lots of different things we could have measured. Keep it simple. I said height, but different experiments would measure different things. Anything you're measuring during the experiment is your dependent variable. And then, of course, you have all the things that have to be the same, which is everything else, and those are your constants. You run the experiment, measure the heights over time, and then you analyze the data. And I'm just going to put this into the notes for now, and we will get back to that. And then you draw your conclusions. Now, that all is a controlled experiment. But something that's really important to understand about science is if you've done all that and nothing else, you've basically not done any science. The next step is critical, and people don't think of it much in science. This is essential. If you do an experiment in your backyard and you don't tell anybody about it, what you're doing is messing around in your backyard. But the moment that you publish your results and other scientists can read it and tell everybody what you did wrong or right, or try to repeat it and see if they can get the same results, at that moment, that information in your report becomes part of the knowledge of what ends up in textbooks. That's how science is made. Everything that we're going to talk about all year, all the stuff that we know, is simply the result of lots of scientists doing experiments, publishing their results, and everybody sort of figuring out which is actually the answer. If you think about science, a lot of times people think, wow, scientists don't know what they're doing. 1 month, they say, butter's bad for you, eat margarine. And then next month, you see an article in the newspaper that says, margarine's bad for you, eat butter, right? The scientists don't know what they're doing. Perfect example during the pandemic. Don't wear masks, wear masks. It's like the scientists don't know what they're doing, right? In fact, what's happening is as new studies come out and are published, scientists read those studies and they get the information out of it, and then they try to repeat those experiments or see if it matches their data, and then they'll publish something, and then another group of scientists will publish something. So it may have turned out with our butter example that The first study was done only with men under the age of 50, and that men under the age of 50 were having a higher rate of heart attacks eating butter than margarine. But then when somebody else redid the experiment, this time with women over the age of 50, it turned out the results were different. And those are both right. They may have done perfectly fine experiments and come up with very seemingly different results, but because their experimental design was different. The people who are wrong here are not the scientists. It's the journalists who are just blaring out a headline, butter's bad for you, butter's good for you. The scientists aren't saying that. They're saying this group of men under the age of 40 who ate margarine had a longer lifespan, right? That's what that experiment is. And science, the ultimate thing that gets put in our textbooks, is a long accumulation of a ton of different experiments and interpretations by different scientists, with things being accepted and rejected and refined. And that is science. And so this publishing data at the end is very, very critical. And that is the scientific method.